Hello, Professor and classmate. I'm Seokhan from Plant Biotechnology Lab under the supervisor Professor Kyun-Oh Lee. Today, I'd like to talk about the green vaccine and glycoengineering. First, what is the green vaccine? It is a vaccine produced by plant cells, not mammal cells. In 2009, when H1N1 flu outbreak occurred, green vaccine was attracting attention, because it was possible to mass production in a short time compared to vaccine production using fertilized eggs. And in 2020, the green vaccine is getting attention again because of the coronavirus. The spread of the coronavirus in a short time, we need a large amount of vaccine in a short time. Vaccine from mammal cells have some riskiness, pathogen infection, expensive equipment, take a long time to cultivate, etc. On the other hand, plant cells have advantages: stability, relatively low price, take a short time to cultivate. However, there are also problems with plant-derived vaccines. Due to difference in glycosylation between species, species-specific residues are generated. The potential immunogenicity of these residues has been extensively discussed in relation to green vaccines. Eukaryotic cells have two kinds of protein synthesis and modification pathway. Like prokaryote cells, protein undergo proper folding and modification in cytoplasm, or proteins involved in membrane or secretions are proper folding and post-translational modification process in intercellular membrane, such as endoplasmic reticulum or Golgi, which have only eukaryotic cells. N-glycosylation is one of the post-translational modification through the uh, through the inner membrane system. N-glycosylation occur at asparagine residue on the consensus sequence motif of N, X, S, or T. X is any amino acid except proline. And 14 oligosaccharide precursor transport to the asparagine residue by OSD complex, oligosaccharide transport complex, and these glycan are important for controlling folding by chaperone gene like CRT, VIP, and PDI. These protein oligosaccharide precursors are initially formed on the outside of the ER membrane by ALG enzymes. After flipped into the ER by RFT1 and then continuous added by ALG enzyme. After the entire oligosaccharide has been moved by OSD complex, it is trimmed with glycosidase and mannose to form mannose 8, glucose 2, and move to Golgi. Since the type and number of glycosylates in Golgi differ depending on the species, the glycosylation process is divided and the glycan structure also differs. In the case of yeast, it becomes a high mannose type, and in the case of plants, it presents plant-specific alpha-1,3 fucose and beta-1,2 xylose residue. And the mammals has an alpha-1,6 fucose residue. These plant-specific residues have potential immunogenicity. Next, what I run. I'm using a CRISPR-Cas9 system to knock out two conserved fucosyltransferase A and B. Vector were designed by inserting guide RNA corresponding to exon 3 and 5 into polycistronic guide RNA region. This guide RNA will bind the sequence corresponding to exon 3 and 5, and Cas9 protein will be cutting this site. I will make a double knockout mutant during master course, like this. This double mutant produces protein without plant-specific residue. The constructed vector will be transformed into Arabidopsis xylosyl transferase mutant to create xylosyl transferase, fucosyl transferase double knockout mutant, and screened using genomic DNA PCR and immunoblot. Furthermore, we will introduce alpha 1,6 fucose to have the form of mammalian cell-derived protein. Last, how can I apply this? In my case, I'm working 경상남도 Agriculture Research and Extension Service Institution. So, as a research worker, I need more academic training. So, through the master course, I want to run biotechnological experiment methods and learn how to write the paper to help in my work. Thank you for attention.